let's say we're going to clone cow. Okay, so the first thing they have to do is collect samples of the cells of the cow. And they can get it just from their skin cells. Let's say we're going to clone a cow. Okay, so we have to, usually it's just easy to get a skin cell. So they get a skin cell from the cow. Let's say this is the cell. This has DNA in it. Okay, this has instructions, directions, what the cell to do. This what's to tell the cell what to do. So this is going to be so we're going to go through cloning the steps. The first step is we have to get a cell from the animal we want to clone, because that we need that information. If we want to clone the animal, we've got to have its instruction manual so we know how to make the animal. All right, so we're going to, let's say we're going to clone a cow. So this is a cell from the cow. Okay, we'll see, this is our cell. I'm going to call this cow A. So this is the cow we're cloning, because we have its DNA in there. Next thing, you can do this with the same cow, but let's just say we don't have to. Let's say the next thing now, we're going to get a egg cell from a cow. Let's say cow B. Okay, so we're going to take the egg cell. So the next step is we're going to get an egg cell from cow B. So this is cow B egg cell, which is called an oocyte. Notice, it has a nucleus in the cell. Okay, so let's show you. This is remember, egg cells, the female sex cell called oocyte. See the nucleus. What we do is actually remove the nucleus. Gone. Nucleus is out. We have an empty egg cell. So remember, if without the nucleus, it doesn't have any directions. It needs instructions to what to do. It's a cell waiting to happen. It's waiting to do something. It needs instructions. It's an empty egg cell. No nucleus, no DNA in it. Okay? So the next thing, they take the skin cell from cow A and the oocyte from cow B. And they use an electric current, so kind of a shock, probably electricity, and that fuses them together. So now what happens is the DNA from cow A is in the egg cell from cow B. So this information for cow A, is because that's the one we're cloning, is going to get inside of the empty egg cell. Okay, and now cow A cell is put inside the oocyte from cow B, and then electric current, like a shock, is applied. So the nucleus from cow A is going to be in cow B egg cell. The cell, it's going to divide and keep dividing, divide, and then pretty soon when it gets to be, it's called a hollow ball of cells called a blastula, that is put inside the uterus of a donor cow. It could be cow C, or it could be a cow A or cow B. And then once it's inserted in the uterus, it's going to keep divide, and develop into an embryo. So it's going to develop into a baby cow. And notice this is not fertilization. There's no sperm involved at all in this process. This is egg cell and using nuclear transfer and cloning. After you get the cell, the animal you want to clone, and in this case we said it was cow A. Next thing, you're going to remove the nucleus of an egg cell from a cow, another animal. You have an empty egg cell from cow B and one skin cell from cow A. So then we're going to inject cow A cell into the empty egg cell. Of cow B. And then I'm going to apply electric shock, some electric current, and that cell is going to keep dividing. And a few days later, when you have a ball of cells called a blastula, this is after a few days, then is implanted into the donor. So the donor is the female that's going to carry the baby, or the surrogate mother is going to carry the baby. After about 9 months, same gestation is like the length of time for pregnancy. So pregnancy, gestation, gives birth to a clone of cow A. So technically that cow C could be cloning itself if we use cow C's cell in the first step. So wherever you originally get the DNA from, that's the one that's being cloned. But cloning has to start off as one cell. So things can't be cloned and they're the adult version immediately. So they start off as a cell. So they have to go through that process of cell division. They have to go through the stages, the embryonic stages, and then be born. So it's not, you can't clone an adult and they're going to be an adult right away. And what is the advantage of cloning? If we think of maybe creating, cows are a big contributor to greenhouse gases and climate change because they produce a lot of methane, which is gas really. They have a lot of gas. So if we can genetically engineer cows that are less gassy, I mean not just give them like Rolaids or something, but just genetically change their DNA so they produce less gas, then we could maybe clone those cows and then would have a less a negative effect on the environment. So that is one possible use for cloning. But if we think of genetic modification, actually most of our processed foods are genetically modified. In fact, 80% of the processed food in their grocery store is genetically modified. Really, most corn is all genetically modified. And they do that because they can create crops that are resistant to pests, so they don't have to use as much fertilizer. And it increases the productivity, so less of the crops are damaged, and it really increases the production of crops.